Okay, well, I guess we're going to go ahead and get started here on lab. And everything is found, as I said earlier, is in the eLearn content section. So lab is, it's not going to track with the content in lecture. It, for whatever reason, the way it's set up tracks separately, but that's okay. It's a standalone class. Now, when we get into AP2, you'll find that the labs and the lecture content track pretty well. So in lab, we have exercises. We'll have unit 1 will be all about the microscope and tissues and stuff like that. And then we'll have an exam scheduled. And then the second unit is going to be bones. And I'm going to give you as much time as I can on that because you're required to learn all of the bones of the body, all 206 of those, as well as the associated openings and processes and stuff like that. And don't worry, because it's a lot easier to do that than it really does sound, believe it or not. And so the third unit then is all about muscles. Muscles are easier because there's only 60 muscles and you've already learned the bones, so you learn where the muscles attach. And then the last unit is probably the most interesting because that's the one where we dissect brains and eyeballs. It's all about the nervous system. And that one sort of does track with what we're covering in lecture. So lab is a 1-credit course. Lecture is a 3-credit course. I will tell you now that don't stress over an A or a B in lab, because the lab grades are not considered when you're applying for any of the healthcare programs, only the lecture grades. I mean, do your best, obviously. You know, getting an A or B is very good, but not at the sacrifice of your lecture grade. You know, you really want to do well in lecture. And if you end up getting a B in lab because you got an A in lecture, that's fine. So don't stress over the lab, but do the very best you can. Now, other things about lab. Meets once a week. If you find you're going to miss a Tuesday lab, there are no other AP1 labs around except the Monday lab. The Monday lab is all virtual. So if you feel the need to attend, you just let me know and I'll shoot you the Zoom link for the Monday lab. So you can go to the Monday lab virtually, or you can always watch the recording of this lab because I'm going to post this in YouTube, just like I'm posting the lecture lecture recording for the day. So you can go back and you can go and watch the lab that you couldn't attend. And you know, sometimes those are going to happen. You know, sometimes you're going to have work events, sometimes you're going to have family events where you just can't get to class. And that's understandable. Things are going to happen no matter what we plan. I mean, none of us, you know, we got reminded very quickly at the beginning of this semester, you know, that no matter how much we plan, you know, events occur outside of our control. It snowed yesterday, and of course, we closed the campus today. None of us expected to be doing a Zoom class today, but we did, but we are. And of course, I'm hoping it doesn't ruin snow days for us. But anyway, the point is that there are ways to make it up, you know, that you can watch it, you can participate virtually, or at least watch it virtually and see what I covered. So it's not the same as being there, but it's pretty close. But if you really want to attend virtually, you can go to the Monday class. I'm fine with that if you need to. Okay, lab grades. There are 4 lab exams. There is no final exam in lab. I'll say that now on the first day of class, and then I'll end up saying that again later on in April. But there is no final exam in lab. You have 4 lab grades, and that's it. You add up your score and you know, 90% of whatever 90% of there's 400 points, 90% of 400 is 360. So if you get if you total 360 points or above, you got an A. And 80% is 3,200 and 320. 320 points. Above 320, you get a B. Simple as that. No extra credit, just straight 4 exams. And once you take a lab exam, you're done with that content. You won't see that content again. So you don't have to worry about going back and looking at your exam and figuring out, why that was marked wrong. It just, you can move on from there. Now, I said in lecture today that I don't know when we're going to finish each unit. So I don't have exam dates listed for lecture. But for lab, it's different. Lab it has to run a pretty tight schedule to get everything done in the amount of time we have allotted. Now, we do have enough time every day, every lecture, every lab day to cover all the content. So we can always start and finish a topic in one day. So we do have set dates for the exams, or we have to track pretty quickly on this. Believe it or not, while this is January the 18th, this semester will be over before you know it. And I've seen this too many times. So, but the topics there do require a certain amount of prep time. When we get to bones and muscles, you will need to spend a fair amount of time learning the bones and then learning the muscles. It won't be something you can do at the last minute. It won't be something you can do the night before an exam. You'll have to spend a fair amount of time learning this, but you don't learn it all at once. You learn a little bit at a time. Same applies to the first test and to the last test. Now, in your content, I have given you a study guide in the lab content. In lecture content, I have a study guide for every exam. Here, I only have a study guide for the first exam, because after the first exam, everything is strictly identification, and you go off and I give you the list of the muscles and the bones and the structures of the brain and so on that you're going to have to know. So there really isn't a study guide for the other exams. But the first exam is so important because it's going to set the tone for the rest of your grades. And a lot of people don't take the first lab exam seriously, and they tank it. I mean, really do really badly because they waited to the last minute and didn't review properly, or just looked over the material and thought they could remember everything. And I'll tell you, after a lot of these tissues all look the same initially. So you need to spend some time getting ready for this first exam. That's why there's a study guide available there for it. Now, having said that, many people that do poorly on their first lab exam end up doing quite well in the course because the first exam sort of gives you an idea of what to expect. Even though I sit here and I give this talk every semester on AP1 labs, and some people don't take it real serious. And that's, and I'll admit, because it sounds boring to say, well, you need to study for the test. Well, of course you do, but a lot of people don't take the course as serious as they should until they get to that first exam. And then that's a wake-up call. So I will also say there are no do-overs in here on the exams. There are no makeup exams. Now, if you're sick and you miss the exam, if you're taking the on-ground class and you miss the exam, then we can work that out. If you are taking the online exam and something happens, we can work that out. Things are always going to happen and get in the way. Kids get sick, parents get sick, you get sick, cars break down, it snows, it rains, it floods. We've seen it all, and they all affect us. We're in accidents. We have to go to the hospital. Grandma dies. Or grandma gets sick and things like that. I will say, the very first semester I taught at Walter State, I had Same one student have grandma die 5 times. I didn't question it, but you know it. So I'm pretty easy when it comes to things like that. But at least give me a reason why you're not going to be there. Not like I didn't feel like coming, or I forgot. It's not a reason. So, but that's the other thing is if you just don't if you don't show up and don't give me any reason, then you know I need something. I can do anything for you. I can do almost anything if you tell me ahead of time. Okay, so enough of that. Let's see what else about lab. When we come to lab on the ground, generally the classroom says no eating or drinking in there, and that's going to depend on the type of lab that we're doing. When we get into dissecting up brains and eyeballs in AP at the end of AP1 lab, then probably you don't want to have anything to eat or drink in the classroom. I certainly wouldn't. If we're just looking at bones and muscles, which are just the models, the life-size models, we're looking at microscopes like we would be today, it's not going to matter. There's nothing there. But if we're ever going to use real specimens, if we're ever going to use any chemicals, then it's a no eating and drinking kind of day. So let's see what else. Again, the classroom is 217. If you're on the ground, it's going to be 217 in Maples Marshall up on the 2nd floor. It can hold 55 students. We only have 24 in the lab. So if you want to spread out, you can. And even the workstations I have around the back wall of the classroom are far enough apart so that you should have good social distancing if you know you're concerned about that. So any questions about what we're going to cover and how we're going to do the lab? I will say that the comment I made earlier in lecture about study groups applies to lab also. Of course, I get that because when you're learning, you know, a lot of this is going to be memory, memorizing the bones, memorizing the muscles. I seriously doubt if anybody's ever going to come up to you and ask you a question about, you know, which objective lens should you use under the microscope at any point in time. You know, so I don't think that's ever going to happen to you. However, I can see real benefit of knowing the bones, knowing all the bones, and knowing all the muscles, and knowing the structures of the brain and spinal cord and the parts of the eyeball. Those are going to have real benefit later on. Some of this stuff, you know, not an immediate benefit, but things like the practical application of bones and muscles and anatomical structures in the special senses and the brain will have some real relevance to you later on. Anyway, anyone have any questions? Okay, good. So what I want to do is we're going to go take a look at first I'm going to explain about the microscope. You may have used microscopes before. I was all set up for lab today. I had the microscopes all setting out since last week, and they'll still be there next week. So we may get some more microscope time in next week to look at slides and stuff, but and maybe make some wet mounts. But today I'm going to talk about the parts of the microscope because you know it is important that if you go into healthcare, that's going to use you end up using microscopes. And you're probably going to be trained extensively in how to use one in whatever program you go into, but it's nice to know your way around the microscope initially. So, but I will also tell you that after we do it in lab, because for today, there's going to be very few opportunities to look at use the microscope as we go on. So, okay, let me get into screen sharing and go into. Here we go, week 1. So you all should see the slide that says week 1 compound microscope and the cell. So microscopes.
I can see it.
Okay, great. Thank you. So there are all sorts of different types of microscopes. Microscopes have been around for about 400 years or more. The earliest ones were simply a brass tube with 2 lenses fitted inside of it. And if you wanted to look at something with greater power or less magnification, you'd have to get a different tube. So different types of microscopes do different things. We've probably all heard of electron microscopes that can magnify things 100,000 times or more. We don't have one. They probably have one at UT, but I'm not certain. They cost. They're real expensive, but they do let you see things, magnified to great levels. Now, what we use is a compound microscope. It is we call it a compound microscope because it has multiple lenses inside of it. And we call it a bright field microscope because we use natural light directed through a device called a condenser onto a slide with a specimen on the slide. We have the compound microscope or bright field microscope stacks a series of lenses so that we have a lens that magnifies the image on the object on the slide, and we have another lens that magnifies that image 10 more times, so that we get a double magnification effect, if you will. The lens that's directly above the specimen is called the objective lens, and then the second lens is called the eyepiece or ocular lens. Now, the objective lens, we have different lenses for different magnifications. The ocular lens is always going to be magnifying something 10 times. So the way our microscopes work is that we have an objective lens that sits directly above the specimen on a slide. A light source passes through the slide and through the specimen and is focused onto that objective lens, which then focuses it into a structure called a prism. A prism bends the light so that we don't have to stand over the microscope and look straight down into the ocular. It bends the light so that it comes out at a 45-degree angle into our eyepiece. It's very convenient for us. 50 years ago, microscopes worked straight up and down, and to look into the study in the microscope, you look straight into it, straight down into it. It was a cutting-edge technological advantage. Excuse me. It was a great cutting-edge advantage or breakthrough when someone decided to put a hinge on the microscope so you could tilt it back, so you wouldn't have to stand over it anymore. Something simple like that hadn't been thought of for like 300 years. So you could actually tilt the microscope. And then later on, it was discovered, hey, if we put a prism up here, prism, is just like a lens that can bend the light rays at different angles. We don't have to do anything. We don't have to bend over it or anything. So anyway, we magnify the image, we run it through the first lens, we run it into the body, what we call a body tube, and then then we bend the rays so they go out through the ocular. Now, when we look at it through a microscope, we have what are called the objective lenses. The objective lenses come in 4 magnifications, and we usually don't say, well, that's 4 magnifications. We usually say X, like 4X, magnifying it 4 times. So, for example, the scanning objective lens has a 4 on it, so it's 4X magnifications, or 4 times. Low power has a 10X on it, which says it magnifies the object 10 times. High power has a 40 on it because it magnifies things 40 times or 40x. And then the fourth lens, which is called the oil immersion lens, has a 100 written on the side of the lens. And it magnifies things 100 times or 100x. And we have them attached to a structure called a nose piece. The nose piece holds all four of these objective lenses right above the slide. Now, our light source comes up from underneath. That was another big improvement, probably 50 or 60 years ago. Up until then, microscopes had mirrors underneath the slide, and the light would be reflected off. If you'd have a candle or a light bulb or something, it would shine onto the mirror, and it would shoot straight up through the lens, through the slide into the lens. And you had to play with the mirror to get, to control your light. And someone got the idea, hey, let's put the light source in the base of the microscope. Another cutting edge, like, why didn't we think of that 100 years ago type of ideas? But it works. You have a light source at the base of the microscope, and then the light is focused. This is another subtle innovation. Light is focused through a condenser through the opening in the stage that holds the slide. Condenser functions like a magnifying lens, like a magnifying glass. The light, instead of just hitting the slide, just like from a regular light bulb, now it's focused. So you collect as much light as possible and aim it right into the specimen so it becomes as bright as you need. So the condenser focuses that light. And we have something called an iris diaphragm that allows us to trim or shade the amount of light we want coming in, because sometimes we don't want too much light there at all. Now, when we do find, we have everything set up the way we want it with the lights on and the slides on the stage, and the lights coming in and hitting the objective lens the way we want it, then we have to focus it. Now, the focusing depends on two dials on the side of the microscope. Usually it's a large outer ring and a smaller inner ring together. Sometimes they're two separate dials. And you generally use the course adjustment to move, you're moving what we call the stage, the platform that the slide sits on. You're moving that up and down. That's how we bring the image into focus. Now, since everybody's eyeballs are going to be a little bit different, you're going to use the course adjustment to get it as close to focusing as you can with that. And then you use the fine adjustment to tweak it so where it is in perfect focus for you. So, like when we're in the classroom and I'm going around and looking at things for you, showing you things on your slide, my eyes are different than your eyes. And so I'll focus it from my eyes, then you'll have to refocus it back to yours. Okay, so this is what our microscopes mostly look like. Now, this is an older version, but it still meets everything. None of our microscopes in Sevierville look like this. All of our microscopes there are a little better, but this is a Morristown microscope, but it has all the same features. So now, first thing the thing we look into is the ocular or eyepiece. And the eyepiece, the ocular, always has a 10x stamped on the side of it, because the ocular has the other lens. The ocular magnifies it, whatever image is coming up off the slide down here, this will magnify it 10 more times. So whether you're looking at something with a 4x objective lens, 4 magnification objective lens, when it comes out here, it's going to be, the total magnification will be 40 times. So your scanning lens, your lowest power, is only 40 magnifications. This is called the body tube. It brings the image from the prism. The prism is located right in here. So see the light rays come up from the light source up through the condenser and the diaphragm into the lens, straight up into the prism. The prism bends it at a 45-degree angle. It goes through the body tube and to the ocular, which gives it one more magnification. This is the nose piece. The nose piece revolves. It holds 4 objective lenses, sometimes only 3, but usually 4. And they're the objective lenses. Now, the objective lenses come in, it's a standard. The lowest power, the scanning power, has a red band around it, and it says 4x. The low power, it has a yellow band on it and has 10x. And the high power has a blue band and it's 40x. So the total magnifications, scanning low and high, the total magnification for the scanning lens is 40. For low power, it's 100. For high power, it's 400. And for the oil immersion lens, it's 1,000. And we probably will never use the oil immersion in A&P, but when you take microbiology, you'll use it a lot to look at bacteria. And this is pretty consistent. Every microscope you ever pick up anywhere are going to have the same colors and the same magnifications on their objective lenses. It just depends whether or not there's a 4th lens on there for oil immersion. Now, this is what's known as the mechanical stage. The way we focus images here is to move the stage up and down. You know, the slide is fastened on the stage, and we move the stage up and down to focus. We can also move the stage left and right and front and back. And that's a real advantage. Again, that probably 50 years ago, the concept of the mechanical stage made moving slides around a whole lot easier, because if you're looking at a slide and didn't have a mechanical stage and you had it just lined up where you wanted it, and then you bumped it with your thumb, and there was a half an hour's work gone to start over again. Okay. Coarse adjustment, and this one is a separate dial. Fine adjustment is a separate dial, but on most of the microscopes that you'll see, you're going to have a coarse adjustment on the outside, and the fine adjustment dial will be in the middle of that. So they're going to be they're going to be on both the left and right sides of the microscopes. It doesn't matter if you're left or right-handed. Yeah. Light source, sounds trivial, but there it is, and it works very well for us. There's our base. The only important thing about the base is if you're carrying the microscope around, you have to have one hand underneath it on the base. And then, of course, there's our light switch. Now, it seems like a trivial point, but every microscope manufacturer puts their light switch in different places. Some put them on the side, so some put them on the left, some put them on the right, some put them in the back. It's important to know where your light switch is, because you're going to be using the same, you're using that microscope. I don't know why they couldn't be consistent and put them all in the same place. We have some very nice microscopes that you'll use in micro, where the light switch is in none of those places. The light switch is on the side of the arm, this up and down piece here, where it not only functions as a light switch, but it's also the dimmer switch. So this is the diaphragm. The diaphragm works much like the pupil in our eyes. It opens and closes to restrict the amount of light or let more light into the slide. We can control that by a little lever on the side here. There's a close-up of the diaphragm. We can, depending on how much light we want in or how little light we want in, because sometimes less is better. We can control that opening. So it functions a lot like our pupil does. Let's see what we see. Okay, there's the mechanical stage. There are spring-loaded clips which will hold the slide in place. And then when we turn these dials that you see here and here, on the far side of the picture, you can move the stage left and right or up and down. You can control very carefully and precisely what you're looking at. Depending on the stage, coarse adjustment, fine adjustment. There's your diaphragm again, and you can just see it at the time here. That's the tip of the condenser up there. You'll see that in this next slide. There's the condenser. Again, it functions like a magnifying lens that takes the light rays coming in from the light source, focuses them onto the objective, to the objective lens right here, so that all the available light goes straight up into the lens. Gives you a much brighter image. Now, these two dials here control the movement of the stage. Again, there's the condenser. And adjustment knob for left and right, as well as front or back. On the microscopes we have in Sevierville, the controls are underneath the stage, so that you don't get your hand, they're not there to get in your way when you're trying to move the slide around. So again, these are much older scopes. I've been using the same picture for 18 years. So I guess these scopes are older than that. So, okay. And there's the mechanical stage that we can, so we have the ability on the stage to move it up and down, left and right, and front and back. So we have a pretty good flexibility there in that. And again, underneath here, you can see the diaphragm here, and you can see how small the opening is for these objective lenses. If you look right into this, this is your low power objective lens. This is the blue one. No, I'm sorry, this is the high power objective lens. That's the 40X. You see how small the opening is. So that's where that condenser that's up inside here really works to put that light right through that little hole there. And diaphragm. Now, so this is a good takeaway slide. This summarizes everything you need to know about magnification. The objective lenses, the red one, scanning power magnifies 4 times. The low power magnifies 10 times. The high power magnifies 40 times, and the oil immersion magnifies 100 times. The ocular always magnifies 10 times. So to get your total magnification, you simply multiply the eyepiece value times the objective value. Now the eyepiece is always 10. The objective lens is either going to be 4, 10, or 40. So your total magnification is 40, 100, or 400. 10 times 40 is 400. 10 times 10 is 100, and 10 times 4 is 40. So that gives you your total magnifications. And of course, if you were going to use the oil immersion, you'd have 10 times 100, which would give you 1,000 magnifications. A couple things you're going to see. The resolving power, we call it resolution. When you look in the microscope under low power, objects may not be very clear, particularly very small objects may not be clear at all, because the 40X magnification of the scanning lens, the 40X total with the scanning lens and the ocular just isn't powerful enough to show you what you're looking at. So you go up to 100, 10X for the objective, 10X for the ocular, and now what you couldn't see before shows up very clearly. Maybe you have to go to 400 to see it, 10 times 40. The ability to distinguish between objects is the resolving power. When you're looking at something with resolution, how well can you see something, with your eye? Is it very clear, or do you miss a lot? Think about this. This is a letter E. This is a letter E that was printed out on a laser printer and cut out and placed under the microscope on a microscope slide. So it's a print out on one of your handouts I sent you. Yeah, so the letter E is pretty small, and that's like times Roman 12. Okay. Now, when we look at that image on paper, it looks very sharp and very clear. But if you magnify that 100 times, you can see it isn't sharp and clear. You can see the splatter of the toner on, or the ink on the paper. You can see it isn't sharp, smooth edges. It's a rather jagged edge and an irregular edge. Our eyeballs don't have the resolving power to see those irregularities. To us, it looks as clear and sharp as the letter E does up here in the word resolution or inversion. These look nice and sharp there. But if we look at them up, magnify them 100 times, you can see that they're not sharp and clear. Excuse me. Anyway, so this is where the microscope helps us see things on the cells. And on the slides we're looking at, we can see structures that we couldn't see with our own eye, with the unaided eye. We see structures at higher magnifications, greater magnifications than we can see at less magnifications. So it's often important to start in the scanning power to find what you're looking at, and then bring the magnification up until you see all the structures. For example, if you're looking at a cell, you may not be able to see the nucleus under very low power. You may have to bring it up to greater magnification to see the nucleus. So if you're looking at bacteria in a micro lab, you may not be able to identify, are they round, or are they oblong, or are they spiral? What looks like a series of dots may actually be rod-shaped bacteria, like little bars linked together. But you couldn't tell that because 40 magnifications wasn't enough. So resolution is very important to us. So is inversion. When you're looking at something, and this has absolutely really, this is an interesting point, but it doesn't, no one's going to come up to you and say, hey, what about the inversion on your microscope? Inversion means that we usually see the object upside down and backwards, because that's what the lenses do. They take it, and remember, you're running the image through two lenses, and so we end up flipping it. So, but it doesn't really matter, because if we're looking at a cell is a three-dimensional structure. It's always in motion. So upside down and backwards is sort of irrelevant, because we're looking at structures, not letters. So, but microscopes will tend to microscopes will flip the image and invert it. So just as an aside, why do we say oil immersion? Well, oil immersion has to deal with what we call the refractive index. When you look at something in water, for example, if you have a drink and a straw in it, you can see that the straw doesn't, when the straw hits the level of water or what, Coke or fluid or whatever, it doesn't line up with the straw that's in the air because the light rays are being bent. Well, refraction is what's happening when we look at structures under the microscope, especially if we've made it what we call a wet mount, a fresh specimen. So light rays will get scattered when a light ray is in a different medium, like water, air to water. Those are two different mediums. Light rays get bent when they go from air to water. We don't get a good image. We don't know precisely where the object is. Like when you're fishing, you see the fish in the water. That's not where the fish is. The light rays have bent the location just enough so that where you're seeing the fish is slightly off where the actual location is. So when we get up to 1,000 magnifications, the light rays are being bent so much by the air molecules around them that they alter the image that you're looking at. Because we're dealing with a very tiny opening in the microscope lens. So what we do to avoid this distortion caused by air is we put a drop of this special oil on the slide and lower the objective lens, the oil lens, into this little droplet of oil. And then the light rays are coming from the light source through the condenser, through the slide, into the oil, and up into the oil immersion lens. And they're not being bent and off in different directions because the air molecules are actually causing the light rays to shoot off in different directions. So anyway, now something else to understand, something a little more practical than refraction and stuff like that. Staining. Most of our tissues underneath our skin all look identical. It's particularly in our internal organs. Most of our internal organs, except for a few places, are very specific. They all have the same, they're all the same color. And it's very hard to distinguish structures. And even if the structures are different, like the liver is dark brown, for example, the gallbladder is dark green. But the heart is, and muscles are red. Everything else looks pretty much the same. So to differentiate between structures, we use dyes. We stain a slide so that we can see, so that different structures stand out. And we've found that different dyes are absorbed by structures at different ways. For example, in microbiology, you'll use a stain known as Graham's stain. Graham stain uses iodine, and it uses a purple dye. And what will happen is, it turns out that pathogenic disease-causing bacteria tend to absorb the purple dye, and we call them gram-positive. And so generally, purple staining bacteria are pathogenic, sort of, and it's not always true, but mostly true. Purple positive pathogenic, whether, the non-pathogenic bacteria tend to stain pink using the stain. And it has to do with the structure of the bacteria, the walls, the cell walls of the bacteria. Other structures in our bodies accept dyes, different types of dyes, in different ways, just like the bacteria do. So we use these stains so that structures will stand out. And the dye allows the light ray to bend and show some contrast for us. So if we were going to, next week, we'll probably, if we're on the ground, those of you that are coming on the ground, we're going to do a wet mount. Those of you that are zooming can watch while we do it. But this is a very simple experiment to show you what kind of cells we have in our bodies, for one thing, and also how to make a wet mount. You take a toothpick and scrape the inside of our cheek with the flat end of the toothpick, smear that on a microscope slide, add a drop of a dye called methylene blue, place a cover slip on there, and place it under the microscope, and it looks like this. It doesn't have the red lettering, but it looks like this. Because the dye, the methylene blue dye, will get sucked up by the nucleus. That's that dark spot there. That's where our DNA is located. And it will also stain the cytoplasm. And it will show you the edge of the cell membrane. So with just a toothpick and a slide and a microscope and a little drop of methylene blue, you can see a lot on your slide. Now, this is what we see in cheek cells. These are human cheek cells. Methylene blue works perfectly for that. And it works every time. This is what it looks like This was a couple of years ago. There's a cheek cell right there, and there's another one, and there's the nucleus there, and here is another one. So even if it's a little fuzzy, it still shows up very nicely. These are what are called epithelial cells. And they're probably the most common cell in our body. They line every opening to the outside. And we have them, our skin is epithelial cells. Our digestive tract is lined with epithelial cells. Our reproductive tract, urinary tract, anywhere there's an opening to the outside is lined with epithelial cells. And the inside of our mouth is lined with many of these, so to protect us. So, and they all look pretty much the same. They're all sort of an oblong-shaped cell with a nucleus. Now, our microscopes are not powerful enough to see anything else in the cell. Even at 1,000 magnifications, we could not see any of the other structures. But we can at least see the nucleus and the edges of the cell. And also we can see the cytoplasm. So it's, as a demonstration, it's a pretty useful tool. So anyway, that is everything under the microscope. Let me get out of here real quick and then bring up the next section on epithelial cells. Here we go. Okay, next thing we'll take a look at are going to be these cells themselves, epithelial cells. Let me bring this up here. Yep. That's still not right on. Okay. I apologize. I had it lined, I had it all set to go. And now everybody should see the one that says epithelial.
I can see it.
Great. Okay. So epithelial cells are probably the largest. As I said, there are more epithelial cells in our body than any other type of cell. There are only 4 types of tissues in our body: epithelial, muscle, connective, and nervous. Epithelial is designed to protect us from the outside. It's also designed to absorb nutrients in our gut, for example. It's also designed to protect us from pathogens and to protect us from wear and tear. So the epithelial cells come in 2 categories. They're either simple, 1 cell layer thick, or stratified, multiple layers. In our lungs, we have simple epithelial cells that are 1 cell layer thick. They are so thin that oxygen can move right through the walls of the cell into blood vessels so that we can absorb oxygen from the outside air passively. The oxygen just simply diffuses. It just passes through the wall of the epithelial cell that makes up the lung wall passes through that into a blood vessel, and we get our oxygen. So very important role there that these simple epithelial cells make. Stratified are where our cells that are there to protect us from wear and tear, like layers of our skin, are stratified epithelial cells, because we're always shedding shedding cells. We're always shedding cells. You're just rubbing your hands together, you can't see it, but thousands of cells are falling off as we do that. But they're there to sacrifice, the ones that are coming off are already dead, but their role is to sacrifice themselves to protect the underlying cells in our skin. The same lining shows up inside our mouth. The lining of our mouth is multiple layers of stratified epithelial cells, because when we eat and drink, cells are ripped off all the time, damaged and destroyed. We don't notice it. That's how you can scrape cells off to make a wet mount. We're scraping off dead cells. Dead cells are on top and they're pushed up from underneath by the living cells, and they die, and more are pushing them up, and they're designed to be worn away, just like on our skin. Every day we're losing cells everywhere. We don't notice it. So stratified protection. Also, the fact that they're so thick, it's very, very hard for pathogens to get in through the skin. In fact, it is a barrier that it's very hard to breach. You can't get a pathogen can't get into your skin if there's no cut or scratch or opening there, because our surface is essentially waterproof, and it's covered with oil that it functions as a bactericide. It kills bacteria. Now, so those are the two categories of epithelial cells. If we look at the types of epithelial cells, the big three are what are known as squamous. That's what those wet mount cheek cell slides I showed you earlier were. Those were squamous cells. Then there's cuboidal cells, which look just like they sound. They're shaped like little cubes. And we have columnar cells, which look just like little columns. So those are the big three. Now there are some outliers here. We have what are known as transitional cells, and we have what are known as pseudo-stratified. It means they're not stratified. They look like they are, so they're false stratification. Ciliated, they have little hairs on them. Columnar epithelial cells. Fortunately, they only show up in a few places. And so they're not like if you look for them all the time. So this is simple squamous epithelial cells. Squamous, same shape as the cheek cells. We find these in the lungs. For example, one cell layer thick, very, very thin. The analogy I used before was single ply, really, really poor quality, single ply wet toilet paper. And even that's probably too thick to really work well as an analogy. But that's how thick, simple squamous epithelial cells are so thin that oxygen can diffuse right through the membrane, and CO2 can diffuse right through the membrane. So if you want movement back and forth without any effort, passive movement, that's what you're going to get with simple squamous cells. Simple squamous epithelial cells not designed to provide protection, because they're going to be worn away too fast, but designed to allow movement from one side of a membrane to another. These are simple cuboidal epithelial cells, shaped like cubes. What we're looking at in here is we're looking down into a series of tubules inside the kidneys. When we send all that blood to our kidneys, because we filter our blood 60 times a day in the kidneys, and we recover 99% of that blood. Because blood is made up of red cells, white cells, and saltwater plasma. We recover 99% of that because we only pee out about 1.5 liters a day. We don't want to lose any more than that. We recover that plasma that we filtered out by letting it move through these tubules and get reabsorbed, passing through these simple cuboidal cells. So all the kidney tubules are made up of stacks of simple cuboidal cells. One cell layer thick. Here's a kidney tubule here, and here's a big one right here in the middle. You know, there's even with the membrane on both sides, they have to go through substances that can pass through here so easily. So in our bodies, primarily, we find these only in the kidneys, in the kidney tubules, because they aid in the filtering process. These are simple cuboidal cells. Simple columnar cells, like this, you can see that they're columns, they're column shaped here. We find these in places where we want to absorb things, like our small intestine. 20 feet of simple columnar cells designed to absorb nutrients. We absorb nutrients, sugars and fats and proteins, and those nutrients pass through this layer into the blood vessels behind them. And so we have simple columnar cells designed to be absorptive. Anywhere we want to absorb lots of something, lots of nutrients, lots of water, lots of sugars, whatever, we're going to have simple columnar cells. We also have an extra advantage here to make, like in our digestive tract, we lubricate everything. We make it easier for food to pass through our digestive system. We have these structures called goblet cells, and goblet cells secrete mucus. Goblet cells release mucus that will trap dust and dirt, and they also lubricate the lining of the this tube here, this opening here. Like this is the small intestine. This is the opening. This is the lumen, the middle of the small intestine. They lubricate this so that the food can go through here very easily before it's absorbed. Here's another example here of some simple columnar cells. You can see how they're lined up. And of course, all these little white things here are the goblet cells, allowing whatever is passing through the middle of the small intestine, the middle of this structure, to be lubricated so that it passes easily. This one is an example of one of the outliers. This is what's known as pseudostratified ciliated columnar epithelial cells. Now, the cilia, these things up here that look like little tiny hairs, are the cilia. Now, the cilia are embedded in a layer of mucus generated by these goblet cells here and here. And there's another one there, and there's another one up here. We find these in our respiratory system so that dust and dirt particles that we breathe in get trapped in the mucus, and then the mucus is pushed up to our throat by the cilia, and then we swallow the mucus. We're not aware we're doing it, by the way, but we swallow about a pint of mucus a day, containing dust and dirt and contaminants and pathogens and things like that, which all get destroyed by the stomach acids when it hits our stomach. So it's a way of cleaning our airway. So we have these respiratory tracts are lined by these pseudostratified ciliated columnar epithelial cells, not just our trachea, but our nasal cavity is lined with these same types of cells. And we also find ciliated columnar epithelial cells lining the inside of the female fallopian tube. because the egg, when the egg is released and moves into the fallopian tube, and, the egg ovulates and is released, the ovary ovulates, the egg is released in and moves into the fallopian tube, the egg has no way to move. And so it depends on being pushed along by these cilia, pushing it down towards the uterus. So that's one of the other places where we'll see these ciliated cells. And here's a better picture of it here. You can really see now they were described as pseudostratified because they look like they're stratified columnar cells because they appear to have multiple layers, but they really aren't. But here are the cilia here. Again, right there's our cilia. This is a close-up on the cilia. The cilia, these tiny hair-like structures, will move together in one direction and then rebound. They'll beat, come back and beat again. So they push things, whether it's an egg, whether it's mucus, they push in one direction and one direction only. Now this is transitional epithelium. Transitional epithelial cells are unique in that they look like columnar cells, but when they stretch, they can stretch. The urinary bladder fills up. Urine collects in the bladder. As it fills up, it gets bigger and bigger. We all know what that's like. As the bladder expands, it stretches, and what looks like columnar cells end up looking like squamous cells because they've stretched out flat. And then when the bladder empties, they go back to looking like columnar cells. And so these are the transitional, because they can go from one form to another. And this is one of those slides that if I didn't tell you what it is, if I put it on a test and you couldn't tell by looking at it what it is. I'll just say that. So this is one of those you need to remember, only because no one's going to come up to you anywhere and say, hey, how about them transitional cells? But you'll be able to pass on your test. And there's a little explanation of transitional epithelium. Lines the ureters, the tubes leading from the kidneys and the urinary bladder and part of the urethra. Here we see them in their relaxed state, where they look like they're columnar, but when the bladder fills up, they will stretch out flat. This is what often throws people a real curve on this first lab test. You see all these cells here, and what are they? Well, some people, it very much resembles a type of muscle cell, and it's not a muscle cell, but it resembles it. But this is stratified squamous epithelial cells. This is what we find on the inside of our mouth. We find this lining portions of our digestive tract. This is what lines the rectum and the anus. This is what lines our mouth and our esophagus, designed to wear away to protect us. Our skin is lined with these type of cells, stratified squamous epithelial. They're designed to offer protection through thickness so that they can wear away and not cause us any harm, and also to protect us from any kind of invasive pathogens. And of course, many cell layers thick. This is, here's the esophagus, for example. This is the stratified squamous right here. Many layers of squamous epithelial cells. Now, some of it can be what we call keratinized. Keratin is the waterproofing compound we have in our bodies. It's the same stuff that makes up our fingernails. And some of the epithelial stratified squamous are waterproof and some isn't. We want the skin, our skin, to be waterproof with keratin. We don't want the lining of our reproductive tract or our digestive tract to be waterproof, because it'd be very painful then. So now let me get out of this. Let me get into this last group here. Now, so those are the epithelial cells, and this last group is the other three types of tissues. We have three types of muscles. We have skeletal muscle, which is voluntary. We control that. It's cardiac muscle, only found in the heart. It's involuntary. It resembles skeletal muscle, but it's involuntary only. We can't tell the heart what to do. And we have smooth muscle, also under involuntary control. It surrounds our digestive system, so it squeezes food through our digestive system, and it surrounds our blood vessels. It allows our blood vessels to squeeze or relax, and squeeze and relax when we're moving blood around. And so skeletal muscle is characterized by having stripes. It's striated. Cardiac muscle is striated too, but it has branches in it, sort of like tree branches. And then smooth muscle looks like a spindle. So this is skeletal muscle right here. These light and dark bands are called striations. And these are the key identifier for skeletal muscle, these alternating bands of light and dark structures. The other thing about skeletal muscle is that skeletal muscle cells have many nuclei. That's what these dark spots here, those are multiple nuclei. They're the striations. There's some fat deposits. We store a lot of fat in our muscle for energy storage. And these are the nuclei. When we are being formed in utero, when we are in the uterus and we're developing, the skeletal muscle cells are formed from many primordial cells, and we never lost the nucleus. But it doesn't matter because our muscle cells don't ever undergo mitosis. Muscle cells don't ever reproduce, so it doesn't really matter. But this is so. Skeletal muscle cells are striated and have multiple nuclei in here. This is smooth muscle. No striations, single nucleus. Again, involuntary. No control over it. We don't tell our digestive system to process that sandwich we had at lunch. We don't tell our blood vessels when to relax and when to contract to move blood around. So single nucleus in here for each muscle cell. And again, this is smooth muscle. Each one of these is a cell, and there's a nucleus in each one. Close up on that. Now, here we see smooth muscle cells in action in the body. This is part of the lining of the small intestine, and there's this outer layer here, the small intestine, and then underneath that layer of tissue is smooth muscle. We have two bands of smooth muscles surrounding our entire digestive system. They squeeze in two different directions to propel food and processed food through the tract from start to finish. The apple you ate this morning will come out tomorrow as waste material, at least the skin will anyway, and the fiber. Thanks to these muscles here squeezing, pushing that through the tract. So here we see two layers of smooth muscle. Now in the small intestine, just like all of our digestive tract, the smooth muscle layers are either circular or longitudinal. One's on top of the other. So we can squeeze in a circular pattern or a lengthwise pattern. Now, in the heart, the only similarity between skeletal muscle and cardiac muscle is that both are striated. That's it. Skeletal muscle doesn't branch. Cardiac muscle does. The reason that cardiac muscle branches is because Every muscle cell in the heart has to contract at exactly the same time with the exact same force every time. Every cell contracts to its fullest every time in the heart. Skeletal muscle doesn't. Only in skeletal muscle, we only use the muscles that we need to do to get something done. But the heart gives its fullest effort every time. The branching is where the cells communicate to each other. There are connections here called intercalated discs. We'll talk all about that in AP2. But these branches here, these branchings allow all the cells to rapidly communicate to each other. They also only have one nucleus per cell too. So cardiac muscle cells only in the heart, they're striated and they look like skeletal muscle a little bit, but they branch and they have these discs here. And the branching and the disc give it away. This is cardiac muscle right here. There's the discs along in here, and you can see how they are striated, but you can see how they're branching off here. That branching allows them to rapidly communicate cell to cell, because the heart has to contract fully every 7/10 of a second. Striations, disc, branches, and nucleus. Now this next area is called connective tissue. Now there's only a handful of structures here that qualify as connective tissue. The definition of a connective tissue is connective tissues have cells in some sort of matrix. It can be a liquid, a solid, or a gel with fibers. So you would have blood. Blood is a connective tissue because blood has cells in a matrix of salt water, the plasma, and it has fibers in solution that help us clot. So even though it sounds a little sketchy, blood is a connective tissue. So the fibers within most connective tissues can be one of these three, or maybe all of these three. You can have what are known as collagen fibers. Collagen is the most abundant protein in the human body. It is amazingly strong. It is very resilient to stress. And they wrap around themselves and give themselves great strength, and they give the tissues themselves great strength. The tendons that form at the end of our muscles, like the Achilles tendon, are made of collagen. Collagen is so strong that it will snap bone before the collagen fibers themselves break. Now, the second type of fibers are elastic fibers. They're made up of elastin, catchy name for elastic fibers. They're made of elastin and they're stretchable, but they rebound. The fact that our skin is flexible and can bend and twist and not get torn or stretched out of shape is because of the elastic fibers present in our skin. And the reticular fibers are the third category. The reticular fibers are sort of the catch-all. Circle that and say catch-all. They form like a net, a safety net. They help hold organs in place. They're sort of the framework to hold our visceral organs, everything like the stomach and the small and large intestine. Why do they always have the shape on a model of along in our abdomen? And why are they held in position? Well, it's because of these reticular fibers holding them in place. Collagen, very, very strong. It runs side by side by side by side. So it's like the collagen fibers run, like if you have one thread, you try to pick up, you have a single thread and try to pick up a 10-pound weight, the thread's going to tear. If you have 1,000 threads lined up next to each other, you can easily pick up the weight. That's what collagen does. They line up together in the same direction, they all pull in the same direction, and they have incredible strength. And when they wrap themselves around a bone, they are as strong as steel. Elastic fibers will stretch and rebound to their original shape. Anywhere where we're going to have movement, we're going to have elastic fibers. Our arteries, the walls of our arteries are lined with elastic fibers because the arteries will stretch out when a surge of blood goes through them, and then they snap back in place. That snapping back is what we call the pulse. If you were to take your fingers and place them on your wrist right behind your thumb, you feel a bumping. That's the pulse. That's the snapping back of the artery in your arm there, what we call the radial artery. As the surge of blood goes through, the artery expands and then snaps back in place. That's thanks to the elastic fibers. Our vocal cords are elastic. We can change the shape of our vocal cords to make different sounds. Our skin is elastic, our trachea is elastic, our bronchi are elastic. And of course, the reticular fibers, as I said, are the framework that hold things together. So here's an example of a connective tissue. This is what we call loose connective tissue. Loose connective tissue has reticular fibers and elastic fibers and collagen in here. And those are fibroblasts. Fibroblast tissues, are making, that's the nucleus to this type of tissue, if you will. There's the elastic fiber, and there is a reticular fiber. And I know that you can look at this and immediately tell the difference, right? No, can't tell what it is. Just take my word for it, what they are. The only thing I can tell you with any certainty is that that is a nucleus and that this is loose connective tissue. Down here we see more nuclei, and here we see a collagen fiber. We can tell this is a collagen fiber because we have parallel structures running side by side in here. So loose connective tissue is the most common of the connective tissues. It's what holds the epithelial cells on the surface to the underlying tissues. And it depends on those fibers to hold, what kind of loose connective tissue it is also depends on the fibers involved here. But this is what we see holding the epithelial, the outer layer, to the layer underneath. Here we see a lot of reticular fibers are very squishy, holding organs together, are very squishy organs like the spleen and the liver. The lymph nodes, for example, don't have a lot of structure. And so they are held in their shape by these reticular fibers, for example. Loose connective tissue right here. A lot of reticular fibers, not a lot of collagen fibers, not a lot of elastic fibers. Imagine a layer of fibers surrounding Jell-O. You have a Jell-O mold, and you have a layer of fibers around it. That's what the spleen's like. It's squishy. Doesn't have a lot of strength of its own. So, dense connective tissue, the tendons and ligaments, the structures that hold our bones to our muscles. Muscles don't attach directly to bones. They attach to tendons, which are made of collagen. Collagen is very, very strong. They have to be to pull on the bone to get the bone to do what the muscle says to do. This is what collagen fiber looks like. Row after row after row of collagen fibers. Dense connective tissue, lots of collagen. This is dense regular. See how the collagen fibers, even though they're sort of wavy, they're still running together in the same direction. Tendons, ligaments, where joints come together, are going to have dense connective tissue. Irregular connective tissue, still dense, where we have more flexibility at a joint. Like we have what's known as a joint capsule, which surrounds like the knee or the elbow. We want to have movement in multiple directions. Cartilage cartilage is a connective tissue. It is collagen fibers, lots of collagen fibers in a matrix that is now a gel. Up until now, we didn't see a gel. We saw, it was a fluid like the cytoplasm of the cell. But now the matrix is a gel in here. So it's called chondrin. Our cartilage is soft and flexible and it has great strength, but it's very, very pliable. The end of our nose is collagen, is cartilage. Our ears are cartilage. Our skeleton starts out initially as a cartilage model, a hyaline cartilage model, and starts transitioning the bone. We have a full skeleton in place, and at 6 weeks after conception, we start converting that to bone. So we lay down our skeleton completely within the first 6 weeks after conception, and then for the next 20 or so years, we're laying down bone. And we have cartilage in our nose, our trachea, and our ears. So cartilage looks like this. Now, something about cartilage is that cartilage lays down its matrix by cells known as chondroblasts. Blast means builder. They lay down their matrix by these chondroblasts. The chondrin is laid down, and then it firms up, and the chondroblast cell gets trapped inside this bubble, and it becomes a chondrocyte. This hyaline cartilage. There is a chondrocyte in a structure known as a lacunae. It's still, it's held in here. It's a living cell stuck inside this matrix of this gel. Now, how's it get fed? How's it get oxygen? Well, cartilage has a bit of a problem because it doesn't have any blood vessels. Cartilage depends on diffusion of nutrients and oxygen to keep the cells alive. The problem with cartilage is that when cartilage gets injured, it takes more time for cartilage to heal than it does for other structures because it gets its blood, oxygen, and nutrients indirectly. Bone is connective tissue. It has the matrix is the hard bony substance. The cells are called within the matrix are called osteocytes, and it's wrapped with collagen fibers. There's the fiber definition. What happens is that we have these osteoblasts, these bone building cells, laying down the matrix called the osteoid, which hardens, forms the lacunae, the bubble, and the osteoblast becomes the osteocyte inside the lacunae. But here we have connections. These little lines here are all canals and tunnels that allow the nutrients, allow blood vessels to reach each and every cell coming off of this central canal here. So bone is most definitely a living tissue. And then each bone itself is wrapped in collagen fibers. So we have the fibers and we have the living cells in a matrix. In this case, the matrix is solid. Adipose tissue is a connective tissue. It stores fat. These are adipose cells that contain fat. There's a nucleus there, and there are elastic fibers and reticular fibers embedded through here to give flexibility and strength. Let's move this right there. Blood, as I said, is a connective tissue. It has living cells, red cells and white cells, in a matrix, which is the plasma, and the fibers are in solution. This is blood. These are all red cells here, red blood cells. That's what's known as a platelet. There's a white blood cell there. The white blood cell's nucleus is visible. Red cells don't have a nucleus. They kick theirs out. So these are all living cells inside the plasma matrix. The last category of tissues, and then we'll wrap it up for today, This is nervous tissue. So we've gone through epithelial, muscle, connective, and now nervous. This big structure here is a neuron. Right here, this is a neuron. The extension off the neuron is called the axon. It sends nerve signals down from the cell body here, down the axon. So there's what we call the neuron cell body where the nucleus is located. These cells also don't reproduce. So all the muscle cells we have now are the ones we're born with. We're not going to make any new ones. We can make them bigger. We don't make new ones. All the neurons we have now are the neurons that we were born with. We don't make new ones. There's the neuron cell body. These little purple dots here are called the glial cells. They do all the support work for the neuron. You know, they provide nutrients, they protect it, they take away fragments of damaged cells, they protect it, they restrict the flow of materials across the membranes of these neurons. So they're very, very busy. There's many, many more glial cells than there are neurons. There's the nucleus right there. And nucleus and nerve fibers. Now, those are dendrites that are connecting to the neuron cell body. Signals come in on the dendrites here and are processed inside the neuron and sent out on the axon. That is what I showed you this earlier. There are the layers of smooth muscle. I should have removed that earlier. Here are the layers of smooth muscle in the small intestine. The outermost covering of the small intestine is called the serous layer. And there's our muscle layers right there. We have a longitudinal smooth muscle and a circular smooth muscle right there. And that is everything I wanted to cover today. So, any questions on anything? Okay, good. I will go ahead and post this to YouTube, and I'll send you all the link for that. And then hopefully on Thursday, we'll have lecture. If you're going to be on the ground, we should be meeting over on campus. So I thank you for your patience today, and I'll see you all later.
I do have one question.
Yes.
So just to clarify, so you said you're going to send us that YouTube channel, you're going to be posting the lectures. Will that just be in an email on eLearn?
Yeah, I'll send you. What I do is I will copy the link and shoot it to you through eLearn.
Sure thing. Just want to make sure.
Yeah.
All right. Thank you.
To it. Okay. Bye.